All right, y'all, welcome back. We are on our last lecture. I want to talk about evolution. We finally got to the good stuff. Well, that's not true, but we got to the good population-wide stuff. Again, I think that you know all of this. I'll move that. I want to remind you a little bit about what you learned in Bio 1 about how evolution happens. Natural selection drives adaptation, but there's a lot of other ways in order that evolution can happen because evolution is just a change in alleles over time. It can be microevolution, which is where we're going to start, meaning that within a species, changes occur all the time, or it can be macroevolution, where evolution occurs for a long enough time and things become different enough that I, as a person, feel like they need a new name. I'm going to put you in two different possible references, and I put this one also in the Canvas site for you. This is a short video about the 5 fingers of evolution. It will help you if you have not learned this mnemonic. It will help you remember all of the mechanisms that we're going to talk about. I tend to use it with my hands even to keep myself on track. But we're going to talk about natural selection here mainly, but we also need to talk about mutations, gene flow, preferential mating, and genetic drift if I want to talk about how evolution actually happens. This set of pictures that I'm using here are from evolution@berkeley.edu, which is an amazing resource for both Hardy-Weinberg and evolutionary practice. If you want to explore this a little further, I really want to send you off into that general direction. If I was teaching a course just on evolution, I would start with this and then branch out in all of the complexities of it. So I think it's a great place to go and review as you take your next level of higher 300 and 400 level bio courses. Natural selection, thumbs up for natural selection, is all about the environment and other living things driving adaptation. This is the major force we think of the frequencies of alleles changing in animal populations. Plants may be a little bit different. They have some weird breeding things that happen that may skew them heavily towards one of the other mechanisms. But this natural selection is the only mechanism that drives adaptation. A lot of other things can change allele frequencies, but only natural selection can make you better at living in your own environment. It shifts allele frequencies within the entire population to favor alleles that provide survivorship and reproduction. It doesn't make things easier, and it doesn't mean that you live longer and longer because evolution is favoring you, your population over time. What it means ultimately is if you live long enough to have babies and those babies survive, you're favoring passing your genes on into the next generation. This was discovered independently by Darwin and Russell Wallace. Lord Russell Wallace and Charles Darwin were in conversations with each other during the 1850s and the 1860s. They agreed that Darwin had actually published the idea first. So when Darwin published his Origin of Species, Lord Russell Wallace actually delayed the publication of his manuscript so that Darwin would get first credit for it. But they independently came up with the same idea. The Wallace-Darwin concept of natural selection is really that individuals vary. There's variation in a population, and that variation is in phenotype or expression of genotype. The variations are passed on. They are heritable. These guys are working at the same time as Mendel, coming from the same expectations that maybe blending might be a thing. They didn't know about genes yet, but they knew that these changes had to be passed on to their offspring. Organisms tend to reproduce too much. Every population makes too many offspring, and some of those offspring, or frankly most of those offspring, don't survive. And really the phenotypes that are best suited to that particular environment at that particular time to pass their genes on are the ones that pass their genes on at a higher rate. Allele frequency changes over time in a population all the time. If the environment changes, then the allele frequency may shift around. Lethal alleles should be slowly removed from a population, but we've seen there's some variation in that. We looked at sickle cell. Those are lethal alleles if you inherit 2 of the sickle cell trait, and yet the environment is actually pushing the allele the other way. We're inheriting 2 dominant alleles, which should be fine, and in the world population is actually the wild type. Those 2 dominant alleles that cause you to make proper red blood cells are a problem in malaria-heavy areas, and so we select for heterozygous advantage. What I'm really teasing at is the idea is we have to consider fitness in the concept of whether alleles are favorable or not. And fitness is just how successful are you at passing your genes on. We talk about gene fitness and offspring fitness, but fitness is basically, can you make babies that make babies? Or can you not make babies that make babies? If we add fitness to this calculation, and we generally represent fitness by the letter W, if I look and want to calculate my individual's survivability and ability to pass their genes on, their fitness, I can look at the fitness W of my homozygous recessive allele, and if it is 0, I really expect that allele to be removed from the population pretty quickly. What this means is anyone who inherits 2 homozygous recessive alleles doesn't pass their genes on. Maybe they don't die, maybe they just can't reproduce, but they don't pass their genes on. I expect very quickly for that a lowercase a allele to be removed from the population. And it's going pretty quickly. It drops precipitously every generation. If A is a lethal allele and inheriting 2 of them kills you, I'm going to give its fitness 0. But if I have a situation where both my having a single version of the dominant allele gives me normal fitness, I have 100% success with my fitness, I'm actually going to keep that lowercase a or that recessive a in my population because they're being harbored in a small number of heterozygous individuals. If you want to calculate this out, and I do not have you doing this on your homework because this is crazy, I can slowly remove lethal alleles from the population, but if there is a dominant recessive relationship over time, I will still see that recessive allele show up occasionally in my population because there are heterozygous individuals that can carry it, and the chances of them running into another heterozygous individual becomes so small that I generally don't actually see the expression of that recessive allele, but it persists in the population. And if you want to really go down a good rabbit hole with this, not doing with rabbits, but if you look up the blue people of Appalachia, there was 2 people that found each other in Appalachia. They came from originally different parts of the world. When they had babies, their babies expressed or failed to express the gene that makes your blood pink and red, and they were kind of bluish. The chances that those 2 heterozygous individuals would encounter each other was incredibly low. The fitness of blue people in a general population is pretty low, not to survivability, but it's harder to find mates if you're blue in historical populations. And so what happened is those individuals, they were hiding out in the Appalachian Mountains. The Appalachian Mountains populations are very isolated. There's a lot of inbreeding. And we actually had this whole family line that was passing along this blue allele because they were breeding consanguinously, very close together with each other. And if your siblings are heterozygous and you mate with your siblings or your cousins, you have a much higher likelihood of 1/4 population of having offspring with that expression of that recessive allele. I do want to point this out. I'm not going to ask you to do this either, but if I want to bring this calculation into and keep heading down into, especially since some of you are taking genetics in the fall again, if I want to actually calculate this, I look at my fitness calculations for homozygous recessive, homozygous dominant, and heterozygous individuals. In this example, homozygous recessive individuals reproduce slightly less frequently than individuals that are homozygous dominant. I can use this formula here that will help me calculate how long that allele will persist in the population. This QG calculation is really the frequency of the allele A at generation time G. This is the starting, what was the original start frequency? I'm going to consider the number of generation that passed, and I can figure out what's the final. If this is 100 generations, I can calculate the final frequency 100 generations later, considering variation in my allele frequencies, or my fitness frequencies. When I do this, I'm varying these numbers basically based on selection pressure. There is a more favorable fitness for individuals who carry 2 homozygous dominant alleles than individuals who carry 2 recessive alleles. If my selection against recessive individuals is very, very high, I'm going to very quickly remove that recessive allele from the population, and it'll become very rare. If my selection against A, my recessive A, is very weak, it's going to take much, much longer for that to disappear from the population, and it may take 100,000 or 1,000,000 generations for any sort of movement in the allele frequencies to noticeably happen. And so I want to come back to this idea of what is selection pressure and what does that mean. What we're looking at here is what happens over time if something is slightly disadvantageous versus very disadvantageous to have. We call that its selection pressure. How fast am I causing the allele frequency in the population to change? And varying these numbers from this is a very strong selection pressure, I'm quickly removing it, versus a very weak selection pressure for this color. It may be found at lower rates, but it's slowly descending, but it's not descending at any measurable part 1,000 generations later, and it's just slightly less frequent. This is natural selection in action. It's just, is it strong natural selection or weak natural selection? There's actually 3 kinds of natural selection, and I didn't color these. These are from your book. So forgive me here. These are red and green. I think we've determined no one's colorblind, though, this semester. My 3 versions of selection pressure and how hard and in what direction against the alleles am I pushing can be described as directional selection. I'm slowly removing an allele from the population. Stabilizing selection, I'm pushing towards a nice medium between my 2 versions of the allele. Or disruptive selection, I'm removing the middleman. And in each one of these, the green is the original population, and the red is what's happening over time as I apply selection pressure. So I want to look at the 3 different kinds of selection pressure and what they actually mean. I have an example from your book that I really like here. These are finches they are talking about. This is just the male and female finch that they're talking about in the text. This is not the variation in beak sizes. The variation that they're talking about in beak sizes is very little. They either get a little bit thicker or a little bit thinner based on environmental changes where they live in the Galapagos Islands. In years where it is dry, there's not a lot of rain. There are fewer seeds available, and so the birds with thicker, bigger beaks tend to do better because they have access to bigger seeds. They're better at breaking bigger seeds open. They can eat the little ones and the big ones, but the big ones are something they don't really compete with a lot of other kinds of birds about. And so we see as dry years happen, we see the next generation has bigger beaks because the bigger beaked birds hard to say, are the ones that have the most successful number of offspring. In years where it's wet or it's normal weather, there's less variation. We see more variation in the beak sizes because big beaks, small beaks, everybody has enough food, so everybody gets to make their babies. In bad, dry years, we see directional push towards bigger and bigger beaks. If the weather changes permanently in the Galapagos Islands and we have a climatic shift and it shifts towards drier and drier weather, I would expect to see over time that these particular species of finch, their beaks get bigger and bigger to deal with eating more and more big seeds, because the ones with the bigger beaks get to have more babies every generation. Stabilizing selection is where the extremes are no good and the middle range is the best. And the easiest way to look at this is looking at birth weights in humans. Being really small, if you only weigh 1 ounce, you're not going to survive. You didn't have time to develop. And if you weigh 43 pounds when you're born, you kill your mother. So we have a very heavy stabilizing selection to be between, 5.5 and about 8 pounds. There's some variation there, but ideally most babies are about the same weight, regardless of how large or small their parents are, because that is the evolutionarily most successful size to be born at. Mid-range is best. Babies who are born at 46 pounds do not get to have babies to pass on those genes to, and babies that are born at 1 ounce also did not get to have their own babies and pass that gene on. Disruptive selection is the one that ultimately can lead to something like speciation. I have 2 extreme phenotypes and maybe some in the middle, but the extremes are selected for. And so over time, I get 2 different types of phenotypes that tend to express with no mid-range happening. I have an example here of bristles on fruit flies. Bristles are their little hairs and how thick their little bristle patterns are on their thorax. Over time, where we have an overlap starting in the first generation, the bristle numbers between all the flies are about the same. Over time, what happens is these flies tend to breed with individuals that have similar number of bristles because they encounter them more often. And so we end up with a population of flies with few bristles and a population of flies with much more bristles. And you see that space in between? There's actually not an overlap there. And if we let this go on long enough, we'll end up having 2 different species. They could fracture away from each other, and we have few bristle flies and a lot of bristle flies as 2 different species from each other, because they're not breeding in between each other. So I want to talk about where alleles come from and other mechanisms besides natural selection. Mutation is our only source of new alleles. If I have a gene pool that is at Hardy-Weinberg equilibrium, nothing changes. My frequency of alleles do not change from generation to generation. If I introduce a mutation or a new allele into that population, that changes my gene frequencies. Now, most mutations don't do anything. If they do something, it's probably bad, but an occasional mutation is a favorable, heritable thing, and that then gets passed into the gene pool. When I want to calculate the rate of mutation, I can calculate a mutation rate. I look at gametes and what is the chances or the frequency of new alleles appearing in those gametes. For bacteria, I can also look in new daughter cells. What is the rate of the mutations that I see in daughter bacteria? Bacteria mutate at a much higher rate generally than eukaryotic organisms. And that's one of the reasons they are so successful at living everywhere. We can also look at evolutionary change through gene flow or migration. Migration is where you move to somebody moves to a new place and you bring your genes with you. When I look at migratory patterns of populations that were previously not breeding with each other but now are, I may bring alleles that mutated in one population but not into another, and then I introduce them across both those populations. Species divide into populations that are isolated geographically, meaning they don't have contact with each other. And then when I open up contact with them, sometimes that can be a source of new alleles. So just kind of fun facts. The B allele for blood type is much more common in Asia than it is in Europe. And in fact, almost to the point in Spain here of having no people who are carrying B. And if you look over here, 30% of the population is carrying the B allele. Um, this movement, and if I start to look and it's showing you 30%, 20%, 10 to 15%, you can see the loss of the B allele is a lower and lower frequency as I move from Asia through Europe. These actually track the Mongolian invasions. So we're actually seeing the remnant of the children of the Mongolian raiders coming into Europe and then leaving by leaving their B allele across Europe. So sort of interesting historical tie-in, but also a migratory population coming in and breeding with the existing population and killing each other. Also, luck plays a role, right? Maybe you were minding your own business and a rock fell on your head. That doesn't have anything to do with natural selection. That's just luck, but you still don't get to pass your genes into the next generation. Genetic drift is basically the effect of luck on the gene pool. And the smaller your population is, the more likely that genetic drift has a big role in what happens from generation to generation. It's just stochasticity or random events. My little demonstration here, I have 10 flowers in my garden. I'm growing carnations. I have 10 flowers, so how many alleles do I have in my garden population for color? There are 10 flowers. These are diploid, so there's 20 alleles. That, I can calculate the gene frequency from that number. My dominant allele, I have 8 out of 20 of them. 40% of my alleles in my population are big A, red, and 12 out of 20 are little a, or white. I have 60% of the alleles in my population are little a alleles. It's a very small population. Random events totally can cause a major change. Like my neighbor's cow, this is Clarabelle. She broke through the fence and she ate my flowers. This has nothing to do with color being favorable for making baby flowers. This was just really bad luck. I chase her away, and then I'm worried about my population now being very small because I actually, in that one random event, have actually significantly changed the frequency of alleles in my population. My big A, red allele is now only 21% of my population's allele frequency, whereas the little a, the white, is almost at 80%. Evolution happened in my flower bed because Clarabelle the cow got out, and she just, because as far as she could reach through the fence. This is just bad luck in a small population, but the smaller the population is, the more vulnerable it is to just random bad luck. And this is a huge problem when we talk about habitat fragmentation and the decreasing size of species numbers. Within a population, if there are few individuals, that species is vulnerable to extinction because it's vulnerable to bad luck. Small populations can occur because of two major effects. A genetic bottleneck. This is a genetic bottleneck example. This is a real thing. It's a cartoon drawing of it, but the New Zealand black robin is a real bird. We think the original population was all different colors. The New Zealand black robin went through a bottleneck event, some kind of illness that actually killed off most of the New Zealand black robins. Here. And the surviving population actually had fewer alleles for color than the original. Color had nothing to do with what killed them, but the end resulting population was very small, and that small population had less diversity than the original population. This new population over time, as these guys made babies and had more and more robins, that red, all those other colors are no longer available because those alleles have been lost. This population is less diverse as a result of an event that happened and made it very small and forced them through a bottleneck. Humans, we think, went through a bottleneck at one point in our ancestry when we were still in Africa that severely limited our allelic diversity. We think that the population that left Africa went through a second very severe bottleneck. I'll show you at the very end, but we know that populations of people that have never left Africa are much more diverse allelically and different from each other than populations in Europe and Asia and in other parts of the world, because they probably didn't have that second bottleneck effect. The founder effect is when I have a huge population and some of those individuals get blown or knocked or migrated over to a new place where they lose contact with the original population. They can't return and breed with them. So whatever made it over to the new little islands or the new location is the only ones that can breed. So in this case, only orange monarchs made it to this island, a variety of monarchs made it here, and only black and white monarchs made it to this island. These populations are very different from each other, just based on luck of who showed up and got to breed with each other. Non-random mating is another way that alleles can change over time. It's a mechanism of evolution. It is when the entire population has a preference for a specific trait. If all of the beetles prefer brown beetles over green beetles, the ones who just happen to be green don't get to make any babies. This is different than sexual selection. I do not have that included in these lectures. This is not males and females selecting on each other. This is when everyone in the population has the same kind of preferences. This can result in a lot of consanguineous or really close breeding with individuals that are an awful lot like you, meaning you share similar alleles and you end up looking at itself. This doesn't in and of itself change allele frequency, but what happens as interbreeding occurs, and there's something called a coefficient of inbreeding, if all the individuals are homozygous, we actually, at some point, we think that the ancestor of all those individuals might have been the same ancestor. Blue eyes is a great example of this. If you have blue eyes, we can trace back that trait to one individual, probably somewhere in Norway or Denmark, that had a mutation for blue eyes. We think that mutation has happened once in human history, and you, if you have blue eyes or are carrying blue eyes, are the recipient of that allele from the same ancestor as everyone else who has that allele. If you have varying alleles in your population, and your population is heterozygous and very heterozygous, the chances that one individual has two alleles from the same ancestor becomes less and less. The less your population is related to each other, the less likely you are carrying alleles from the same individual. You're not inbred in a nice way. If I let this go on long enough, I can drive speciation, the idea of creating new kinds of things. Species is a bit of a difficult thing to define, but basically we define it generally in biology as when we get to the point where we think something needs a new name. A lot in biology, they use what's called the biological species definition, that if individuals can breed and make babies and grandbabies, and they choose to do so, we say they are the same species. If they choose not to or cannot make babies with each other or grandbabies, then we say they are different species. The process of going from one population into two different distinct things that cannot breed with each other is called speciation, or a divergence of species. Now, I think in Bio 1 you went into a whole series of ways this happens. I'm just going to highlight them. I'm not going to do that whole lecture. But you might want to go back and look at your notes about the mechanisms of speciation. I want to look at this from more of a genetics point of view, where we're looking at genetic divergence between different species over time. And this can happen in two ways. I can have my ancestral population here, and my ancestral population, if there are no significant changes in its genome, over time, this species persists. Anagenesis is when I have this population, it's not just one individual, but a whole population, and within this population, mutations and genetic drift and bad luck, good luck accumulate until this individual is so different and time is passing from this individual that we feel like they probably could not have bred to each other, and we give them different names. They're distinctly different from each other. these two species have formed because time passed and they became very different. That's called anagenesis. Cladogenesis is when this ancestral population now, the one we just made, experiences some kind of separation where one population goes off in one direction genetically and one goes off in another direction genetically, and they separate from each other into two distinct novel new species. That's called cladogenesis. Cladistics is the science of making branches. They make a branch in the tree or the phylogeny of life. When I draw this, ignore the little circle here. When I draw this, remember that these are populations that are changing over time. This population can change just to random luck or mutations, but at some point during cladogenesis, my population gets separated by either a physical or a not physical barrier, and that causes my two populations to diverge into different directions. As time passes, these populations become so distinct and different from each other that they actually become unable to breed, unable to make babies, unable to make grandbabies, and now, according to the biological species concept, they are different species. So what makes them split? Well, if you're using the biological species mechanism as your guide to what is a species, it's reproductive isolation that keeps two species apart. Now, reproductive isolation can occur when I have a physical barrier that is separating a population. That could be that a river appears between two populations, a road. We do this a lot in habitat fragmentations. It could be some of them sailed off on India as it headed north during continental drift. That's called allopatric. Allo means different. Patric means country. It's a different country separation. Alternatively, there's sympatric speciation. Sim is the same. Patric is still patriotism, where something other than a physical barrier, a preference, a mating preference evolves. I end up shifting breeding times. All of those things can lead to sympatric speciation. It's a less tangible barrier. And I think these two words really have their roots back in the history of biology, where we thought that all species speciated because of a physical barrier initially. We thought everything was allopatric speciation. By the early 90s, that had shifted, and we recognized there's a lot of things that can happen besides a physical wall. So we divide those into two separate things. Once they split, they are kept separate by not being able to make babies. These can be prezygotic things that make you not make babies, meaning you can't even make a zygote. This is prezygotic. These could be living in different ecologies, having behaviors so different that you don't want to mate with each other or don't even recognize that you're the same type of thing. It could be being alive at different seasons. It could be literally mechanical separation where you can't get your genitalia together, or it could be physiological, where you may not be able to make your gametes recognize each other, so sperm and egg won't form a zygote. All of these are prezygotic. I've reasoned here this is temporal or seasonal. This temporal means time. Or these barriers could be postzygotic. You could fertilize the egg and make a zygote, but that may never grow up to an adult. That would be about survival or viability. It can be about fertility, where you make a zygote, but that zygote grows up to an adult but can't make babies, like a mule. Or it can be the weird one, hybrid breakdown, which really occurs primarily in plants, where the hybrid is lovely. Take two species and breed it together, I get something amazing. But if I try to breed that baby with others like itself, I try to make an F2 generation, it can't. They don't survive. How fast does this happen? Well, it can be really slow. Darwin hypothesized that evolution is slow and gradual, and it can be really, really fast. Stephen Jay Gould, who, if you're looking for summer reading, wrote a series of books and articles about the weird stuff involved in evolution. The Panda's Thumb is a great start to that. The book, The Panda's Thumb. But also, pandas have 2 thumbs, they have 6 fingers. Stephen Jay Gould hypothesized that some species actually change in rapid bursts, and then they stay constant for a very long time. The difference between these 2 models is really understanding the history of what happened on planet Earth. Gould worked with a lot of fossils. He was looking at the history of time and our understanding of the evolution of creatures over time on planet Earth, where Darwin didn't know that. We think that there are bursts of change and then times when things change gradually. Gould's idea is called punctuated equilibrium. Change happens in bursts. Darwinian evolution suggests that it is gradual, and it's probably something in between these 2. So I know a little bit about modeling evolution. I realize this last lecture is very long, but they really tossed sort of an entire other course at the end of this, but it's important. When I model evolution, I model it using phylogeny. Phylogeny is a hypothesis about how evolution actually occurred. And I can model this by drawing a phylogenetic tree. I look at my root or where my ancestors came from, and then every time I have a branch or a break where I divide that population into 2 different things, this is saying there is some kind of evolutionary event that separated my population. Sympatric, in the same country, or an allopatric, a different country situation, where part of the population went on to destiny of being species A, and the other part of the population went on the destiny to being the ancestor of B and C. These edges, it says tips, they are also called leaves, or if you're feeling really spunky, in most trees, unless it says otherwise, the ones at the leaves or the tips of the tree are generally extant. That's the opposite of extinct. It means that they exist now. When we make a phylogeny, we try to build it as a monophyletic tree, one ancestor leading to all of their descendants. Any other way of doing it, you're making a mistake. Here is the ancestry of some vertebrates here. This common ancestor is the common ancestor of lungfish, but also this ancestor, this is the ancestor of amphibians, mammals, reptiles, and birds. So what does this tell you about birds? You all already know this. Birds are reptiles. They actually come from the dinosaur lineage. I don't know. I have one sitting on my knee right now being super cute, but earlier she was just running around tearing stuff up. They definitely feel their ancestry some days. We use molecular clocks to figure out the rate that this is happening. A molecular clock is really a gene that we can predict evolutionary events on. Often it's something that doesn't change the function, but it has a pretty predictable rate of developing a mutation that we can measure by looking at sequencing of the genome. Molecular clocks there's many different molecular clocks, and you're going to pull one for your research based on how divergent your species that you're looking at are from each other. We hypothesize that mutations occur at a pretty regular rate over time, and so we can use the appropriate molecular clock to measure how long ago did these 2 species separate from each other. Our understanding of our ancestry all the way back to reptiles and fish is based on a series of molecular clocks. That's where we get those dates from, and fossils. Our human evolution is complex. I suspect all the other animals are too. We understand a little bit more about human mutations and evolution because we're terribly interested in it. This is a human migration map. It's based on data from the Human Genographic Project, which is a project originally started by National Geographic and is still using mitochondrial DNA and other DNA to try to trace how humans moved around the world. We have the highest diversity of alleles and genetic variation in African populations that never left Africa. There are populations in Africa where the diversity in one village is more diverse than all of Europe and Asia combined. We lost a lot of genetic diversity when we left Africa. We probably went through a bottleneck. There are a few survivors were the only survivors that continued on that migration pattern. We know that Homo sapiens originated in Africa in Ngorongoro Park. We have evidence here of early hominids and the first few footprints from the Laetoli footprints of humans. The first few steps that we have to look at of humans walking and doing very human things. The human evolution is insanely complicated again. When we look at our genome, we see evidence of other species genes in our genome. It's not always sort of straightforward as we had originally thought. When I look at this population, this is the ancestor of the genus Homo, which is Homo devisonians, Homo neanderthalis, and then this is Homo sapiens, this group right here. When we look at populations across this is extant people, so modern humans, we see, if I genetically sequence you, that you have a certain amount of Neanderthal or Devisonian DNA inside of you. I don't have a great answer to how that mechanism is. We know that we interbred with other species, which makes the biological species concept hard to use in this case. And we know that we probably kept some of those favorable genes. You're not like 50% Neanderthal, but if you have red hair or you have really fair skin, we think that those are genes that were brought from the Neanderthal genes. If you're interested in this, I do link a couple different papers towards the end here, this original article, about how we actually understand this by looking at the X chromosome. The first Homo member of the genus Homo to leave Africa was probably Homo erectus. They made it far into Europe, and we have populations that were living in Britain and then eventually were wiped out. Homo neanderthalis left Africa later and migrated towards Europe. Most of the Neanderthal settlements that we have excavated are in France or in Germany, hence their name. They're probably a little bit different from us, but not noticeably enough for you to be uncomfortable, I would think. Not like, it wouldn't be like a gorilla sat down next to you on the bus. This would be a person that was slightly shorter than you, and we think with a slightly squeakier voice. We have found fossil evidence of hybrids between Homo sapiens and Homo neanderthalis. So not only genetically, but fossil-wise, we know that we bred willingly and often when we encountered them. But if you look at most Europeans, you have about 1 to 2% of your DNA is going to be Neanderthal in origin. So down here, if you click down here, this is a little bit more accessible than this as an article to read about how our Neanderthal, and then more recently we've discovered the Devisonian link. And we seem to be, like many other species we have found, an amalgam of some of the species that were really, really close to us. Nope, there's no wrap up. I want to go back to... Goodness gracious. So with that, try that again. So with that, I'm going to wrap it up. I don't have a practice with me because I put them in the homework. We've been practicing Hardy Weinberg for a while now. There are a few in the homework that are a little bit, I don't know, probably excessive. I want you to look at them with your eyes from learning all the genetics you have learned. I am asking you to work through one of them that asks you to take a gene sample out of an imaginary fish population and do everything from PCR it all the way through to actually read the genetics. I'm really interested in seeing how that works for you. Let me know if it's making sense. I'm asking you to read the trees. That's why you have 3 tries to get everything right. But use the hints. They actually, all but one of them are fairly helpful. One of them I have no idea what it's talking about. And give yourself a little bit of extra time with that. And then I will see you in the lab We have a little bit to wrap up, and then the last week is full week is really all about your presentation. So ask me if you have any questions on that, and if not, I'll see you in lab. And that's the last lecture. You did it. All right. I'll see you soon. Bye-bye.